Hello, everyone. My name is Samra Turajlic. I'm a consultant medical oncologist at the Royal Marsden NHS Foundation Trust in London, and I lead a research group named Cancer Dynamics at the Francis Crick Institute, also in London. Well, of course, I'm delighted for this recognition for our team, really, not for me, from ESMO. It takes a village to deliver a translational research program. It's very much an interdisciplinary, multi-team effort. So on behalf of our team and our collaborators, of course, I'm delighted by this prize. Yeah, thanks for asking me these questions. So in the context of the TracerX renal study, what we tried to resolve is this incredible clinical diversity that we see in patients who are diagnosed with clear cell renal cell carcinoma. Diversity in metastatic efficiency, diversity in clinical outcomes, whether it be from surgery or from treatment. And yet, on the other side of that clinical diversity, are very simple genetics. So this has bewildered all of us in the field for a number of years, and made us think in the TracerX Renal Consortium that perhaps by applying an evolutionary framework of understanding, we could reconcile that diversity of clinical phenotypes. And essentially what we found is that clear cell renal cell carcinoma is characterized by highly conserved patterns of tumor evolution. And what I really mean by that is that there are repeatable paths by which genetic mutations occur in a particular order or in combination, and it's those conserved patterns of ordering and combination that produce different evolutionary tempo, different evolutionary outcome, and a different clinical outcome. So this is really the first time in any solid tumor that we have an evolutionary classification that can predict patient outcomes. So for us, the next step is really to try and bring this into the clinic, and we're working on different methods by which we might implement this information in the clinic. You also asked me about the adapter study. So this is a prospective clinical trial, a phase 2 trial of nivolumab in clear cell renal cell carcinoma. Trials like this are normally primarily focused on the outcomes such as response rate, progression-free survival, and overall survival. But we were really determined to make biological endpoints a main part of this trial. And the reason is that we have this conundrum in clear cell renal cell carcinoma. It's a highly inflamed tumor, more than any other solid tumor type. And yet that inflammation doesn't pertain good prognosis like it does in all other solid tumors. It essentially associates with negative prognosis. And that made us question as to whether the immune cells inside these primary tumors were really tumor-specific at all. And in order to account for all different possibilities, how nivolumab might be acting to produce benefit in these patients, we did a multitude of downstream immunological assays, and the place where we found the signal was TCR sequencing. So what we discovered is that in patients who benefited from nivolumab, before starting treatment, there was already evidence of expanded T cells, expanded TCR clones, so specific antigen-primed clones, which upon binding to nivolumab expanded further and became cytotoxic, and that's how they impacted the patient outcome. In patients that didn't benefit from nivolumab, there was a paucity of these T cells, and once we applied nivolumab, the T cells that were there were replaced by novel TCR clonotypes, and these patients did not benefit. So this tells us two things, really. One is that at least some of the immune infiltration in clear cell renal cell carcinoma is tumor-specific, and two, the key to the mechanism of action in nivolumab in this disease is binding and expanding this particular population of T cells. Thanks for that question. So at the start of the pandemic, we were compelled to set up this study called CAPTURE, really because the pandemic had revealed a huge knowledge gap in the interaction between cancer, immunity, and infection. And all the decisions we were making about cancer patients, whether we advised them to self-shield, whether we interrupted their cancer therapy, were really just based on an assumption that patients with cancer would be more susceptible to COVID-19 or more susceptible to severe COVID-19, and what's more, that the therapies that they're on could make this vulnerability even worse. And there was a long list of potential therapies that could be implicated in this, and all of these are life-saving therapies. So we were very reluctant to continue with this attitude without really generating some evidence. So that's why we set up the study, and the findings of the study, where we probed both T-cell and B-cell responses initially after infection and then after vaccination, were that in patients with solid cancer, the immune response was largely preserved and associated with age more than the cancer factors, so what type of cancer they had or what type of treatment they were on. Unfortunately, patients with hematological malignancies didn't fare so well, and their immune response was often blunted, either because of the type of malignancy that they have or because of the treatment that they had received. These data and other data generated around the world had helped to prioritize patients with blood cancer for further doses of vaccine. But even recently, as we examined the immune response to 4 vaccine doses, it seems that there is a proportion of these patients that remain highly vulnerable, and we should be aware of that as oncologists. Now, relative to your question about ongoing evolution of the virus, I think this has relevance to the entire population globally, not just to patients with cancer. And clearly it will be important to continue monitoring for emerging new variants, because what we've and others have shown is that the likelihood of evasion by variants relative to vaccines that patients have received in the past has been more pronounced with the most recent emerging variants. So this is certainly something that we'll need to keep an eye on and we need to respond to. But I think it will be a population level response rather than only applicable to cancer patients.